Good afternoon, dear students. Today, Histology department represented for you the lecture Blood, Lymph, Reticular Tissue. At the beginning of our lecture, we make something discussion about what is a tissue and how the modern histology operate. This is terms of the modern histological knowledge. So that the tissue is a system, assemblage of cells and cells products that have common structure, origin and functions. That's definition to take in from the old phrase of Xavier Bichat, the one of the famous histologists and founder of histology like a science. He was described 21 types of tissue, and to use tissue like terms, word for description of the level of tissue organization. He was writing that in a living organism, the cells assembled into the tissues which resist to death. And modern histology to have to discuss this question for component of the tissue like system of cells and cells product. Example, extracellular matrix ordinarily contain ground substance and fabric component. Another, this is non-cellular elements, symplastas, syncytium, and post-cellular elements, ordinary derivatives of cells. What is the symplastas and syncytium we will have to discuss later. But what's the main idea? That cells characterize common structure, originate from one source, and to use effectively same functions, and produced some products. Like example, except extracellular matrix, we have post-cellular elements. Ordinary, that cells which lost the nuclei, or maybe some elements of cells. And symplastas and syncytium, it's a result of two variants of different behavior of cells during the differentiation. From one side, symplastos. This is a result of fusing of more than 10, sometimes 100 cells, and to form common cytoplasm and multinucleated protoplasm. And from another side, syncytium, it's a result of not full completed cytotomy. So during mitosis, it's not divided fully because between the cells is present intracellular junction like intracellular bridges. Through these bridges, the cytoplasm migrated from one cell to another. Of course, my dear students, this is disorders of cell theory, because we know that each cell is separated from another cells by the cytoplasmic membrane. In this case, that's position, this is postulate, is broken. From another side, of course, symplastas, multinucleated cell, is not a cell, it's also non-cellular elements, because we have to classify this as elements like products of cells differentiation. And another moment, this is specialized tissue. We will discuss in the practical classes epithelial tissue, nervous tissue, but first of all, we have, after a control point, about embryology and cytology and methods. It happened for division LA first division. We'll have next week. First division students have control point next week. Another after 2 weeks, maybe 2-3 weeks, yes. But in different case, first practical classes after embryology and control point will be topic which is named epithelial tissue and tissue level of organization. But we have no time for made lecture for this, and just very fast, I was telling you a minute ago all definition of the tissue like a system. Next, this is very important theme, the theme which is named blood and the tissues of inner medium. The famous Russian scientist Alexei Zavarzin was investigate the connective tissue, lymph and blood during many years of his life, and the result of that he was writing, inside human body existed a assemble of tissues which derives from mesenchyme and contain high quantity of extracellular fluid and different cell types. This is tissue of inner medium. The main functions of this tissue is regulation of homeostasis. Homeostasis we will discuss a little later, but the main functions of this tissue, blood, connective tissue and lymph, is regulation of homeostasis. Both of this tissue derives from mesenchyme. What is the mesenchyme? Let's next slide of our lecture is shown to you the mesenchyme, microphoto of original slide. That slide you have seen it in the ordinary practical classes for topic connective tissue, but you have seen here a lot of cells, irregular shape. These cells has very good visible inside the cells nuclei, and also have seen dividing cell. This is a metaphase. Also, these cells doesn't characterize the fibrous component surrounded, but we have the big spaces which maybe contain extracellular fluid or extracellular matrix. Is this tissue mature? No. That's one of the primordial gem level of development of tissues. But many scientists too decided the mesenchyme it's primordial progenitor tissue or embryonic connective tissue. Sometimes it's like that, like embryonic connective tissue, because mesenchyme give rise to the formation of different types of tissue. We will study it in the embryology. From mesenchyme is arise the loose connective, dense connective tissue, vessels, like example, endothelium, and smooth muscle cells of vessels and smooth muscle cells of internal organs, all derived from mesenchyme. It's a very difficult question about the mesenchyme like a primordial gem tissue, and we can't be named the mesenchyme original tissue because it lost the fibrous component absolutely. So But from these mesenchymic cells, as you have seen, like aggregation, we was taking this picture from the Russian book. It was made famous Russian histologist Afanasyev. And we have seen here the formation of vessel, the primary blood vessels arise from mesenchyme in the wall of the yolk sac. This is specialized primordial structure which arise from mesenchyme. Look, outermost cells to transform into the endothelial cells of vessel capillaries, and inner cells will transform to the blood stem cells of blood. We will discuss this question for continuing topic hemopiosis. Another type of tissue very resemble on mesenchyme. This is reticular connective tissue. What is the common features of reticular connective tissue and mesenchyme, and what is the difference? That's both of this tissue is named Fales syncytium. Why is named syncytium? Because they form the meshwork structure from one side. From another side, why is named Fales? Because we very good seen under the electron microscope the junctions between cells, reticular cells in reticular tissue, joined by some desmosomes, interdigitation, some another types of intercellular junctions, and because that is named Fales syncytium. Original syncytium can be present only in spermatogenic epithelium of seminiferous tubules in the testes. All another type of meshwork structure, like mesenchyme, like stellate reticulum, for example, and this is classical reticular tissue. This is Fales syncytium. From other side, we have seen that between this is reticular tissue cells, very good visible clusters of lymphocytes. What does it mean? It means this is lymphoid organs. Is really true. This is a picture from the structural organization of lymph node. And you will have seen in the practical classes about blood, second slide, which is named reticular connective tissue in the lymph node. What's the differences with mesenchyme? In reticular connective tissue, except cells also have seen their fibers. In this picture is not visible, but when you have investigates the original slide, you have find it. What's the functions of reticular connective tissue? Reticular connective tissue is also specialized type of connective tissue which provide the creation of niche for stem cells and provide the surrounding medium for hematopoietic cells, lymphopoietic cells and myelopoietic cells. Especially the main source of this reticular connective tissue present in red bone marrow, the stroma, the structural base of red bone marrow, also contain reticular connective tissue, but also in other cells they are present. And the difference of that, some lymph node, spleen, consist in the stroma, especially reticular connective tissue. Today we will study reticulocytes, and many students have mistaken for what's the full name of cells. Remember, please. So in the reticular connective tissue cells we name reticular connective tissue cells. But the unmature red blood cells, unmature erythrocytes, has also name very resemble reticulocytes. But this is reticulocytes is the cells in blood, not in reticular tissue. Thank you for this, for your attention for this part, and we have back for discussion of blood. Look at this interesting picture. In this slide, we have the illustrations of interactions between some very important organs of blood and blood system. Blood system is not cardiovascular system. It's also not a lymphatic system. But blood system is combined that lymphoids and hematopoietic organs like red bone marrow, spleen, organs which provide hematopoietic and supporting functions like liver, because the liver too provides the hematopoiesis from the 5th month of prenatal life, and also, sorry, from the 5th week of prenatal life. The spleen also, and red bone marrow, this is the main hematopoietic organ, medullary hematopoiesis providing. And all these organs is joined with blood through the vessel. Okay, so in volume of the human adult organism, the blood, it's approximately average 5-6 liters. The heart pumping the blood into the blood vessels. The red bone marrow generate a new population of blood cells, all types of blood cells. Liver, that's gland, exocrine glands, which to synthesize the plasma proteins. All plasma proteins except immunoglobulins, gamma globulins, except this is gamma globulins or immunoglobulins was made in the liver. And spleen. Spleen, this is an organ for rare circulation of the blood cells and symmetry for many types of blood cells, for erythrocytes, for thrombocytes, platelets, and any white blood cells is also can be die via apoptosis inside the liver, and after that replacement by another new renewal populated cells. But the renewal population, this is a function of stem cells in the red bone marrow. So all this system is provide the normal function of blood. What is the blood? Blood, in Latin word sanguis, in Greek hema, full definition. Blood is a fluid, connective tissue that circulates through the cardiovascular system and composed of the fluid intercellular matrix, which name is plasma, and formed elements consisting of the various types of blood cells as well as platelets and erythrocytes. Why? But the platelets and erythrocytes lost a nuclei, so they are post-cellular form elements, is not a cells, without nuclei. So the main definition you should be writing in your practical lecture notes. Blood is a fluid connective tissue that circulates through the cardiovascular system and composed of a fluid intercellular matrix plasma and formed elements consisting of the various types of blood cells and platelets and erythrocytes. Well, next we just watching the components, how does it happen, ordinary in laboratory. In laboratory, we have taken the biopsy of the blood to put this blood in the laboratory tube, and we have to see that if start the centrifugate, we have centrifugate, rotated this many, maybe sometimes 10 or maybe 20 laboratory tube in the special centrifuge device. We have, as a result of central action of centrifuge, we have to disseparation the blood into the some fraction. Look, the biggest fraction will be the red blood cells. These post-cellular elements to accumulate at the bottom of the tube because it contains iron in hemoglobin, specialized protein, specialized pigment, and specialized component of the red blood cells, and it consists around 40–45% of all blood. From another side, we have seen between the red blood cells and plasma. Plasma will be upper portion. It's very light because it doesn't contain some cellularic elements. It's an extracellular fluid, extracellular matrix of blood, plasma. And we have that plasma is consists around 55–60% of the blood after centrifugation. And also we have white blood cells. Less than 1% of this is fraction. By the way, look, in book of Jangueira, basic histology, we have seen here the special hematocrit. That's hematocrit. It's a relationship between the quantities of formed elements, both red blood cells, white blood cells, and plasma relationship. We will discuss a little late, but you need to know that of course it's different between the condition of the blood and between the two humans genders like female and male, because the quantities of erythrocytes is different. Why we discuss also a little late, and just look, this is very important, that cubic milliliter of blood is equivalent to a microliter. All complete blood count value in the discuss for special hemogram we will have to writing in your practical book a little late. Look, and white blood cells, which is sometimes is named Buffy cord, because located between plasma and erythrocytes. Buffy cord consists from platelets and leukocytes like neutrophils, lymphocytes, monocytes, eosinophils, and basophils. But you have seen that any of these cells contain granules, which is named granulocytes, like neutrophils, eosinophils, basophils, and another under the microscope is not invisible, these granules. This is monocytes and lymphocytes. Originally, granules is present, but in light microscopy don't identify it. Well, so let's now talking about functions of blood. Protective function, one of the main for human body. Blood is involved in defense reactions such as phagocytosis. This is the functions of many leukocytes and monocytes. immunity, inflammation, regulation of inflammation, and blood clotting. You can ask, what about inflammation? Inflammation is adaptive reaction against external invaders. And of course, we have specific reaction like immunity and maybe another non-specific defense, like we have talking about phagocytosis like that. And of course, blood clotting, that's formation of preventing the bleeding via the activities of some plasma proteins we will discuss, and platelets like main component of blood for that. And second, more important function, respiratory functions. Respiratory functions mean transmission of oxygen and carbon dioxide between the tissue. Some oxygen migrated through the blood into tissue, and carbon dioxide migrated from the tissue into the blood. Trophical functions, trophy, transmission of nutrients, different nutrients. Sometimes it's amino acids, sometimes it's lipids, polysaccharides, some another. Excretory functions is connected with deduction from organism of the various waste products as results of the life activity. During the catabolism, it sometimes appear very danger products like urea, ammonium, creatinine, and some poison like bilirubin, hemosiderin, and some others, poisons and pigments which need to be inactivated. So excretory functions, also functions of blood. Humoral function, a carrier of hormones and other biological active agents. Of course, that endocrine system can be worked, and more there, the intercellular interactions can be activated without blood-like agent for carrier of these substances. And homeostatic functions, what just we have to discuss. That's homeostasis that maintains constancies of inner medium of an organism. Like example, what does it mean? Maintains constancies of inner medium. Regulates the osmotic pressure. If we have to something eating, something drinking, sometimes inside our organism it migrates acids, some alkalines products, maybe disorders of redox level. As effect of that, we have specialized system in the plasma and specialized cells in the blood, which to provide these homeostatic functions. We discussed during this lecture all these questions. So, started from the plasma. That's components of plasma. They dominate in the substances water. Water is around 93% of the plasma. Organic substances 7 to 10%. Proteins 6 to 8.5%. Other substances, lipids, polysaccharides, hormones, enzymes, and non-organic substances, bicarbonates, around 1%. Because that we have bicarbonates, it prevents the free radicals activities and decrease the proton levels. Like the concentration of proton, you need seen here 7.36 level. It's neutral, is not acidic and not very alkaline, so we have to see in that neutral level. And osmotic pressure, the resemblance of the blood is resemble on the fluid of sodium chloride physiological solution. Its concentration of sodium chloride 0.9% in the water. And we have to replace for any special physiological and injection procedures. We need to have to use the fluid sodium chloride liquid with concentration 0.9 for the regulation, the replacement of the blood for the very short time maybe. And all injection we have to use during that time with physiological solution base. So now back for discussion of components and function of plasma proteins. So plasma proteins can be divided on the some fraction. This is a light fraction, 60% albumins. Albumins, it's a light fraction of proteins which have been produced by liver, carry the hormones and some other organic substances, but the main function of these proteins is regulation of osmotic pressure. Regulation of osmotic pressure. Heavy fraction, 40%. It's globulins and fibrinogen. Globulins can be classified into alpha, beta, and gamma. Gamma globulins, this is a famous immunoglobulin, so antibody, to provide the deactivation of free antigens in the reaction of opsonization. You will study in immune response. But from another side, we have the fibrinogen. Fibrinogen and prothrombin, one of the main proteins of plasma which provide the blood clot formation. More fully, you have studied the blood clot formation in physiology and biochemistry. And look, we just discussed, like examples, some functions of globulins. The relative volume of the blood cells in plasma is about 40 to 45% and 50 to 60%, respectively, for between two humans' sex, like genders, like female and male. Male get it more level of hematocrit, and female less, 40-45. This value is called hematocrit. Hematocrit, this is relations between relative volume of blood cells and plasma, between plasma and blood cells. Hematocrit, definition. Was writing now, yes? What about examples of globulins? The very famous is transferrin. Transferrin is carry the iron. That's iron can be taken, the iron from the food. But we also have ferritin, which to take in the some irons after the erythrocytes die, after the process of death of erythrocytes, agglutination of erythrocytes in the spleen especially. But we have discussed a little late. So ceruloplasmin of one of the proteins gamma globulins which to carry the cuprum, and also very important for the regulation of intercellular process. And transcobalamin to carry the vitamin B12. Transcobalamin play very important role in the anemia when happen some disorders with vitamin B12 and sometimes disorders in the digestive tract, where is this vitamin B12 it's absorbed and regulated through some cells. And transcortin. This is an example of proteins which carry the corticosteroids inside the blood. What is a serum? Serum is a plasma of blood without fibrinogen, and prothrombin. After centrifugation, ordinary it's a yellowish liquid. So serum to play very important role for the treatment. Look, that when blood leaves the circulatory system, either in the test tube or the extracellular matrix surrounding the blood vessels, plasma protein react with one another to produce a clot, which includes foam elements and pale yellow liquid called serum. Serum contains growth factors and other proteins releases from the platelets during clot formation, which confer the biological properties very different from those of plasma. From elements, we start our discussion from the postcellular elements, erythrocytes and platelets. Both these elements derive from cells via higher specialization, which is also named differentiation of cells. The mechanism of creation of erythrocytes is named karyorrhexis, or extrusion of nuclei. So that's happened inside the red bone marrow with the participation of specialized cells, the macrophage, which is named feeding cells in the erythropoietic tissue. So from another side, we have seen the platelets. Platelets, it's also specialized cells, but if erythrocytes carry the gases, the main functions, oxygenum and carbon dioxide. The platelets, two main functions, this is blood clot formation, participation in the agglutination in some steps of blood clot formation. The mechanism of formation of platelets is demarcation of the giant cells, megakaryocytes. We have to discuss after some minutes. And look, if this is cells lost the nuclei, is not cell, is postcellular elements, the leukocytes is a true cells, can be classified in granulocytes and agranulocytes. We was telling about a minute ago, but we have to continue our discussion with the original slides. Look, this is microphoto to illustrate it for you how is visible the blood smear under the microscope. After procedure of preparing of blood smear, we have seen a numerous red blood cells. Erythrocytes is very easy to identify because lost a nuclei, without nucleus. Sometimes it's visible like empty area, like empty spaces inside, but this is only shown like optical effect, but of course inside the cells nothing else present except hemoglobin. and plasma membrane. In other cells which have been seen here, this is white blood cells, but you have seen it's absolutely another. And for identification, this is type of cells, we use specialized stain, Romanovsky and Gimza stain, or sometimes in the European countries name Wright and Romanovsky and Gimza, Wright and Gimza stain. But both methodics is the same. We use a special basic stain Azure second and AOZ like acidic stain. Look at this. We have seen the cells which is named neutrophils. And this is neutrophils is very good identified between other cells because it gets the multilobulated nuclei. It's divided on some segments, and pale cytoplasm, but contain two types of granules. One granules is azurophilic, another is oxyphilic, because is named neutrophils. From another side, we have seen the cells with a big nuclei. This is lymphocytes, is agranulocytes, lymphocytes. And another cells also have seen numerous segments of nuclei, 1, 2, 3, and a lot of granules in the cytoplasm. Basophils, ordinary stain azure, azurophilic granules, because is named basophils. Basophils, neutrophils, is also we have find here and cells of eosinophils late. But look at this one, we have seen very interesting particles between the cells. This is platelets. In the practical classes, you necessarily watch these slides, different microscopic slides, and have to compare the normal and pathological condition of blood. Well, so what is the hemogram? Hemogram, the number of blood cells in certain blood volume is called hemogram. Each liter of blood contains erythrocytes for male 4 to 5 in 10 and 12 per liter, and for female 3.9 to 4.7 in 10 and 12 per liter. Of course, you can ask why between male and female we have different levels of erythrocytes quantity. That's result of adaptation during a million years of human evolutions. First of all, is result of more developed muscle in male, and another, this is results of different lifestyle between the female and male during the prehistorical periods. So we discussed that question in the practical classes again, but how is working now in modern time? During this million years, the hormones of male and female start to regulate the condition of hemopiosis. Example, this is action of one of the important hormone, estrogens. That's estrogens, it's inhibit the hemopiosis because the quantities of erythrocytes less in female. From another side, we have the leukocytes. Leukocytes, this is cells of white blood cells. It contains around 4 to 9 in 10 and in 9 per liter. So these cells less than red blood cells and or erythrocytes. Also, we have seen platelets. Platelets 180 to 320 in 10 in 9 per liter. So you need to remember that this is a level, volume of cells. You should remember an answer for each practical classes and maybe control points especially. Except the cells level, volume of cells, we have the some another diagnostical features like color level. This color level is identified the quantities of hemoglobin from 0.85, 1.15, velocity of erythrocytes adhesion for male more, middle like 10 millimeters per hour, but female from 2 to 15 millimeters per hour. And it also depends from different functional conditions and localization the person in the physical levels of the planet. Like example, it's the changes in the mountains and in the valley. So hematocrit 40-45 for female and 50-60 for male. Next, we're talking about erythrocytes. Erythrocytes are the most numerous cells in the peripheral blood. Each drop of blood contains about 100 million of them. Erythrocytes are produced in the red bone marrow and their lifespan approximately 120 days. It circulates in the blood till the erythrocytes not die in the spleen. We have seen in here electron microscopic micrograph, very good visible wall of the vessel, its capillary wall, endothelial cells, epithelium, simple squamous epithelium of blood vessel, and we have seen here the discocytes, that shape is like disc. This is erythrocytes, of course, but the shape with like disc, and biconcave disc. And also we have seen platelets here. Next, when baby is born, the quantities of red blood cells, it's around 6 to 10 and 12, and 9 in 10 and 12 per liter. And all men also have approximately same results, 6 to 10 and 12 per liter. That's the changes of the level of erythrocytes with the age changes. And what is it? What's the name? The decreasing of quantities of red blood cells or erythrocytes. This is named erythropenia. Ordinary erythropenia is result of hemolytic poison or toxins. If we have some snakes bites or maybe some toxical effect after bacterial invasions and some others, also we have to identify the erythropenia if this is toxins or hemolytical poisons destroy red blood cells. Increasing the quantities of red blood cells is named polycythemia, erythrocytosis. So we have double variant. The first name is polycythemia, and another name, same, is analogous, or erythrocytosis, increasing the quantities of red blood cells. That's condition, polycythemia, possibly primary, if we have talking about myelopoietic disorders, extraproliferation, myelopoietic disorders, primary polycythemia, and secondary, during diseases like heart failure, liver disease, lung disease, and erythropoietin disorders. Look at this picture. This is a photo of blood smear of a patient with a polycythemia. The quantities of red blood cells, erythrocytes, more than normal, more. And you can to identify it if we investigate under the microscope. Erythrocytes are biconcave disc 7-8 micrometers in diameter, micrometers, and in inner diameter 0.8 micrometers. And the highest of these parameters is 2.6 micrometers. This is unical shape of erythrocytes considerably enlarges its surface area for gas exchange. Erythrocytes are extremely elastic and deform radially when passing through the smallest blood capillaries. What about hemoglobin? Because this is like container. Erythrocytes is container of hemoglobin. The while erythrocyte cytoplasm is filled with pigment inclusions hemoglobin. Erythrocytes transport oxygen from the pulmonary alveoli to the peripheral tissue and carbon dioxide from peripheral tissue to the pulmonary alveoli. In the lungs and region of the higher partial pressure of oxygen, hemoglobin proliferatively pick up oxygen to form oxyhemoglobin in the lungs, to form oxyhemoglobin, which is high affinity to oxygen. In the peripheral tissue, a region of the highest partial pressure to change in carbon dioxide. Oxyhemoglobin releases its oxygen, exchanging into the carbon dioxide to form carbonyl hemoglobin. But we also have carb hemoglobin. Carb hemoglobin, it's pathological. It's happen when carbon oxide migrated inside, joined with hemoglobin. That's maybe result of death, mortem effect, if not help for person. So you need to separate the oxyhemoglobin, carbonyl hemoglobin, and carboxyhemoglobin, or carb hemoglobin. Okay, next now, what about another functions of red blood cells? This is participation in cells for the ABO blood types. You have seen here the agglutinogens and agglutinins, which is present agglutinogens on the surface of red blood cells. Agglutinins is present in the blood plasma. So we not discuss fully today, but how is joined with cytology of erythrocytes. That's a plasma membrane. It's a site for this discussion. Erythrocytes are surrounded by the plasma membrane. In the glycocalyx of the plasma membrane, the A and B responsible for the types of blood is present. That's a specialized receptor which carry the agglutinogen is named glycopharins of blood types, agglutinogens. In other type, this is special proteins, which is named band-3 proteins, band-4-1, and ankyrin actin. Look now to see this specific shape of the cells. Red blood cells, it's supporting by the special protein which is named spectrin. Look at this one. Spectrin to form very intensive fiber and microfilaments. And this is spectrin, ankyrin with joint spectrin with glycophorins, to form submembranic components of red blood cells. Internal surface of membrane contain these proteins: spectrin, ankyrin, band 4.1 proteins, actin, and that's cytoskeleton of red blood cells to support the shape of these cells by concave disc, and with the time of degradation of proteins, especially spectrin, start some changes of shape. Also, plasma membrane of red blood cells, the cytoplasm, to transport it of amino acids and polypeptides. Types of shape of red blood cells after the senescence and during some disorders. The classical shape of erythrocytes, diameter 7 to 8 micrometers, which we discussed a minute ago, is named normocytes. Normocytes. That's if the red blood cells less than 7 is named microcyte, 6 micrometers and less. If the more than 8, macrocytes, more than 8.5 micrometers. And the condition of blood during which is best possible seen polymorphic variations in diameter is named anisocytosis. So anisocytosis is a functional condition of blood which characterize the polymorphic variations of cells in diameter. I mean red blood cells. And ordinarily it's happened in anemia. If we're talking about example, that is visible like microcytosis. During iron deficiency is visible microcytosis, microcyte. And during the deficiency of vitamin B12, it's happened macrocytosis. So, how is visible under microscope? We have seen the tissue, this is organs, this is blood capillaries, and we have seen how this is red blood cells is aggregates like the dimes or maybe coins inside. That's phenomenon is named Rolex. And ordinary erythrocytes not aggregates like this. This is also functional characteristic. During osmotic pressure, we have changes of osmotic pressure. The red blood cells also lost the biconcave shape. It's possible during some hypertonic solutions. We're talking about the plasmolysis, and hypertonic solutions, plasmolysis, yes. So we're talking about another moment, this is poikilocytosis. Poikilocytosis is another pathological condition. Sometimes it can be described like functional condition. Polymorphic variation of red blood cells in shapes. Look at this one. Any cells, it's got the dome shape, like dome shape. This is named stomatocytes, dome-shaped cells, like this, for example. This is outer and this is inner. And also we have seen the planocytes, flattened shape. Spherocytes, globe shape. Echinocytes, this is spine shape. And sickle cells, it's like during the sickle cells anemia, like pathology, we have seen after a minute example. Look at this one, is poikilocytosis, example of like process we identified in the blood smear. Look, this one is echinocytes, also may be present spherocyte, like these spherocytes, and some other like sickle cells also visible. This is pathology, of course, and sickle cells anemia characterization. This is a phenomenon which is present in the blood, peripheral blood of some African nations. In Africa take place, this is paradox, but that's changed the shape of red blood cells, of course, decrease their oxygenative functions, because we're talking about anemia. This is dysfunctions, one moment, cooperate with resistance to malaria, and this is people not suffering from malaria. So very important for them, but is needable the treatment for normal blood oxygenation. Reticulocytes. Just we a minute ago talking about what is the difference between reticular cells and reticulocytes, and now you need to know the full name of reticulocytes, like erythrocytes type. It's around 1 to 5%, in peripheral blood sometimes to 10%, which is named polychromatophilic erythrocytes. Immature erythrocytes. In cytoplasm contains many organelles like Golgi complex, rough endoplasmic reticulum, ribosomes. It can be identified by supravital stain methylene blue. Look at this one. We have seen very beautiful picture. This is a classical blood smear, and we have seen how the granules of and cistern of rough endoplasmic reticulum and Golgi complexes identified in these cells, with difference with another which is normal, and in higher magnification also very good visible how that structure change, different type of reticulocytes. And under the electron microscopy, very good visible that inside this is reticulocytes, is visible dactyosome of Golgi complex, mitochondria in cytoplasm. This is also Golgi complex. So it's unmature red blood cells or unmature erythrocytes. Platelets. Platelets are not cells. They are the cell fragment derived from megakaryocytes in the bone marrow. The lifespan of platelets 1 to 2 weeks. We have seen it here, like elements. And also in the bloodstream they destroy, they migrated in the bloodstream, and after 2 weeks they destroy in the spleen. Platelets measure around 1-3 micrometers in the size. In the blood smear they appear in round or oval particles. Megakaryocytes. This is the giant cells which is never migrate from the red bone marrow because they produce the platelets. Look at this one. We have seen megakaryocytes with many nuclei, a lot of nuclei, multinucleated cells, and we have the components of cytoplasm separated like platelets. Here also visible nucleus of megakaryocytes and the component of cytoplasm which is separated via demarcated lines to platelets formation. Ordinary, this is demarcated lines, is result of a long rough endoplasmic reticulum. In cytoplasm, these cells is separated. Megakaryocytes. If megakaryocytes occasionally will appear in peripheral blood, This is one of the diagnostic phenomenon which indicates of destruction of red bone marrow. Ordinarily happen in some like causes, for example, like a cancer of the red bone marrow. Platelets morphology or structure of platelets. Platelets are surrounded by the plasma membrane and has invagination connected with a tubular system inside, which probably functions is sequestering calcium ions. This is invagination to provide the sequestration of calcium ions. Platelets has been divided on the two portions. The first is granulomeria. Granulomeria, look, this is a part which with a lot of granules inside, and hyalomere. Hyalomere. So classified each of them. Granulomere. The granulomere stains purple in the blood smear, is composed of alpha granules, dense bodies, occasional mitochondria, lysosomes, clusters of glycogen particles. Functions of granulomere. The alpha granules are 0.3–0.5 micrometer in diameter and contain platelet thromboplastin, special factor. The dense bodies are 0.2–0.3 micrometers in diameter, contain pyrophosphate, ADP, adenosine diphosphate, and ATP, adenosine triphosphate, serotonin, and calcium ions. What about hyalomere? Hyalomere stains faintly in the blood smear, is a reach of microfilaments and microtubules which just beneath the plasma lemma and encircle the periphery of the platelet, maintaining its shape. Look at this one, it's the functions of blood clot formation very fast. We just to see a lot of factors of blood clot, and we not study in histology fully, you will study it in biochemistry, for example. Platelets are important for the blood coagulation and blood clot formation. When the wall of the blood vessel is broken, platelets adhere to be ruptured and to the vessel, which is named the agglutination of platelets. The platelets will aggregate into the platelets clot and the site of vessel injury and will release other substances like serotonin and thromboplastin outside. Effect of it, we have the functions of serotonin and thromboplastin. Serotonin, a potent vascular constrictor, vasoconstrictor, serotonin, causes the vascular smooth muscle cells to contract, terribly reducing their local blood flow at the site of injury. Thromboplastin initiates the conversion of fibrinogen to insoluble fibrin. It's a very important role after the calcium ion waves initiated this process, and the formation of fibrin clot. This reaction requires the presence of calcium ions, of course, and disorders of platelets. First of all is writing now thrombocytopenia. It's pathological decreasing of quantities of platelets. Another variant, opposite, thrombocytosis, increasing of quantities of platelets. In electron micrograph, we have seen the image of platelets under the scanning micrography. Okay. Leukocytes. Leukocytes, white blood cells, contain a nuclei and organelles in their cytoplasm and can move like an amoeba. Leukocytes can circulate in the blood for several hours and leave the blood through the wall of small blood vessels, venules, to enter the connective tissue where they perform their specific functions. Leukocytes participate in the defense reactions of the body, such as immunity and inflammations. Example, immune functions provided by lymphocytes and monocytes And inflammation sometimes is combination of lymphocytes and leukocytes, basophils for example. Leukocytes are subdivided in two groups, granulocytes and agranulocytes, depending on the presence or absence of specific granules in their cytoplasm. Look at this picture. It's very good visible that the granulocytes can be classified into the neutrophil, eosinophil, and basophil, according to the affinity of granules for the basic, acidic, or both stain. As we have seen, the eosinophil is more staining by eosine, but the basophil more staining by azure. And one moment we have seen the neutrophils is both of them is staining as basic and acidic stain. Both this is granulocytes characterize multilobulated nuclei. Another group is agranulocytes, lymphocytes and monocytes. The difference is in the shape of nucleus. In the lymphocytes, nucleus ordinary round shape, but in the monocytes, the nucleus is like bean shape or maybe horseshoe shape, but without some lobules like in the leukocytes, granulocytes. Well, so leukocytes. In human blood, quantities of leukocytes 4 to 9 in 9 per liter. One moment we also have seen that increasing of leukocytes is named leukocytosis. Leukocytosis. Ordinary leukocytosis take place in infectional disease and inflammations. Physiological leukocytosis after feeding very often take place. Leukopenia, the decreasing of leukocytes, it's a functional condition, decreasing of leukocytes in blood. Ordinary leukopenia take place after radioactive diseases. These cells provide non-specific defense. The leukocytes provide non-specific defense, but except lymphocytes. And for the identification of diagnosis of condition of blood, we need investigates the blood smear under the microscope and calculates how many types of white blood cells, leukocytes, lymphocytes, can be visible in each blood smear. Example, we have seen the parameters of leukocytic formula. It's quantities of leukocytes per liter relatively in the percentage for it. Example, the maximum quantities, it's segmented nucleated neutrophil, ordinary 47, 72%. 72%. Step neutrophils, ordinary also named band neutrophils, 0.01 and 0.06%. That cells, it's less mature than segmented multilobulated neutrophils. Another metamyelocyte is young, very young neutrophils, ordinary 0 to 0.01%, I means around 1%. And this is less than 1%. Also named metamyelocytes. So all these cells is different stages of differentiation of neutrophils. But we have to identify it in the blood smear during some disorders. Like example, shift the leukocytic formula to the left. The left corner, we have seen basophils, ordinary parameters 0 to 0.1%. This is basophils, which can be identified very seldom in the blood smear, very seldom. If it's increased, it's shown the high level of inflammations. And eosinophils 0.05 and 0.05%. We have to see it when the inflammation decrease ordinary. So if we're talking about shift the blood smear leukocytes formula into the left, it's signaling inflammation, but it's a stat identified with increasing of stub neutrophils or metamyelocytes or basophils and eosinophils, not segmented neutrophils. Agranulocytes, ordinary lymphocytes 18 to 37%, and monocytes 3 to 11%. In this case, if we have to shift to the right, we're talking about oncological problem, because increasing of monocytes especially and lymphocytes to show the activation of immune system after the oncological problem. So look at this. First of all, we discuss the neutrophils, and this is your examination electron micrograph, which is represented for us the major neutrophil. That's lobulated nuclei and numerous granules. Any granules is more dark, any is more light, and this is one of them is a specific granules and non-specific granules. That's a specific granules also dense and more around the azurophilic granules is named. Like here is more azure stain. And later the number of the extremely electron dense, we have to identify it in electron scanning transmissional electron microscopy. So a definition of neutrophils. Neutrophils are the most numerous leukocytes in normal human blood. The lifespan of the neutrophils is less than 1 week. Neutrophils are round cells 12 to 14 micrometers in diameter. The nucleus has 3-5 lobules connected to each other by the thin bridges. It's very good visible here. Also, cytoplasm contains 2 types of membrane-bonded granules, azurophilic granules and non-specific primary and specific secondary granules. The azurophilic non-specific primary granules are lysosomes. Lysosomes is more dense granules. In addition to various hydrolytic enzymes, they also contain the enzyme myeloperoxidase. This is a special identification of neutrophils. That's enzymes, myeloperoxidase, producing the bactericide molecule of oxygen from hydrogen peroxidase. And Ilya Mechnikov, when investigate the phagocytosis of neutrophils, to name the neutrophils microphagocytes. And this is example of image of band cells. So in band neutrophils we have shape of nuclei like questions or maybe like S shape. So next, specific granules of neutrophils. Specific secondary granules comprise about 80% of the granule population, are small, 0.1, 0.2 micron, and they do not stain are not visible under the light microscope. They contain bactericidal substances, lysozyme, phagocytic, collagenase, and alkaline phosphatase. This is also very important enzymes. The neutrophil's function is a phagocytosis because that's alkaline phosphatase is present and collagenase. So remember this because it's very important for answer the topic about the cells. Neutrophils form the first line of defense against bacteria. They migrate from the bacteria's bloodstream to other connective tissue. Neutrophils first adhere to the bacteria and then engulf them in the membrane-bound phagosome. Phagosomes fuse with their primary lysosomes and their enzymes kill the bacteria. That's the name microphagocytosis. Then secondary granules release their bactericide substance into the phagosome, digestion of microorganisms, but some neutrophils die during that process. The accumulation of dead bacteria and neutrophils and tissue fluid constitutes is formed the pus. But there is a great need for neutrophils in the body, the cause of infection and inflammation. Bent neutrophils increase in number during the beginning of inflammation. This phenomenon is called a shift of leukocytic formula to the left. Look at this one. This we just have seen the two mechanisms. Receptor-mediated phagocytosis of neutrophils, bacteria, and another, this is respiratory burst. The two types of mechanism, receptor-mediated phagocytosis and respiratory burst, to provide the killing the bacteria by the cationic proteins and lysozyme. So next is migration of neutrophils through the wall. This process is named diapidasis, extravasation. During this process, we have seen the stages of it. Ordinary, like mast cells, is release some heparin, histamine, and factor of cytokines, neutrophils, neutrophils taxis factor. Taxis is movement. So then mast cells release some histamine, heparin, and this is factor of neutrophil taxis factor. They start to chemically invite the neutrophils for this area. And neutrophils, to feel these chemical substances, hemokines, to start to firstly rolling, after that integrin activations, adhesion, capture and adhesion, after that joined with integrins, spreading, and after that start extravasation. After the migration into the tissue, phagocytosis of bacteria take place with the special cytokines TNF and interleukin-1. Okay, structure of neutrophils. In forensic medicine, so often can use the neutrophils for sex identification with the bar bodies in present around 3% of female. Immature neutrophils have rod-shaped nuclei and the name-band neutrophils under the normal conditions around 5% of the smear. Next, electron micrograph to show for us that's also cells. You have understand this is also neutrophils because that's a multilobulated nuclei with stalks is visible here and small granules. We have continued our discussion. Basophils. Basophils are round cells around 8 to 10 micrometers in diameter. The basophils has a segmented nucleus that is masked by numerous large basophilic granules. Very often is not visible nucleus. The specific granules are large, 0.5 to 1.3 micrometers in diameter, and membrane-bonded spherical structure. They contain heparin, histamine, and peroxidase. That's a heparin. It's ordinary, if histamine, it's vasodilator, and provides the leakage, the plasma outside the vessels. So heparin, it's substances which prevent the extravasation. It's ordinary heparin increase the movement of blood inside the blood vessels and an agonist of histamine. These granules stained metachromatically with a mixture of the dyes using the stained blood smear, primary as eosinophilic. What does it mean? Metachromasia. That's an example when the conditions when the granules change the stain color, ordinary on opposite, from the blue to the red color. That's happened only with basophils. Basophils regulates the tissue homeostasis and contributes to inflammations by releasing the histamine, heparin, and the eosinophil chemotoxic factor. The basophilic plasma membrane contains immunoglobulin E receptors. When these receptors bind the immunoglobulin complex specific antigen, the basophils degenerate and can initiate the allergic reaction and degranulate and activate the allergic reaction. Extremely serious case, anaphylactic shock. We have seen here classical basophils, but we not have time for discuss more properly. You have to read in the text variant. And we have also two variants of basophils with different conditions of granules. Look at this, this can be identified. And basophilia. This is condition when quantities of basophils increase more than ordinary. Ordinary take place in allergic reaction. Eosinophils are round-shaped cells around 10 and 14 micrometers in diameter. The eosinophilic nuclei has 2 or 3 lobules. Ordinary electron micrograms to show the special elongates crystalline core in these granules. We have seen that contain special enzymes histaminase, destroy histamine, and peroxidase, and major basic protein with the amino acid arginine. That's eosinophils inactivates histamine, releases the tissue basophils inflammation sites by histaminase. Eosinophils are implicated in activating of killing parasites. Though parasites is dying after the activities of crystalloid has antiparasitic functions. Eosinophils are believed to phagocytose antibody-antigen complex 2, an allergic reaction of parasites and vengeance cause an increase in the number of eosinophils in the blood. So, classical electron microgram, for exam, you have seen the eosinophils with lobulated nuclei, and also we have seen numerous specific granules with the crystalloids in the center. Okay, again we have identified eosinophils. And eosinophilia, that is pathological conditions after the increasing of eosinophils. So we have maybe talking about, in this case, parasitic invasions. Lymphocytosis. Lymphocytosis are the round lymphocytes. Lymphocytes is round small cells, 8 to 10 micrometers in diameter. Lymphocytes have a round dark nucleus that occupies most of the cell's volume and narrow rim of the right blue cytoplasm, like here we have seen it here, blue cytoplasm. 3 groups of lymphocytes can be identified according to the size: small, medium, and large. In the bloodstream, most lymphocytes is small and medium. Look at this one. It is small, so this is a medium lymphocyte. Ordinarily, we have seen the nuclear yard, and we have numerous mitochondria, some organelles in the cytoplasm. In ordinary light microscopy is not visible. That's one is the effect of appearance of the lymphocytes. As you have seen how the lymphocytes can stay back into the lymphoblast. This is a reaction of blast transformation, specific reaction in the lymphopoiesis. And this is small lymphocytes for comparison of previous. And large lymphocytes, diameter 10 to 11 micrometers. Immunological classification of lymphocytes you can study in the practical classes in our text variant, like T and B lymphocytes, and B lymphocytes give rise to formation of plasma cells. Is not lymphocytes, but is a product of lymphocytes, which is occupy the connective tissue. The description of the cells you can find in the text variant of our lecture. T lymphocytes ordinarily provide the functions like regulatory cells, T helper cells, and effector cells, T killer cells, and T cells memory, T memory cells, and null cells or natural killer, which is not reagate, not need the some activation through the T helpers. All in other materials you have find in the text variant. We look at this one, lymphocytosis, increases of lymphocytes, ordinarily taken in the some oncological process, and monocytes. Also we have seen the shape of like bean-like shape, but we discussed before, and I want to show to you the last photo. This is types of monocytes in peripheral blood. You have to identify it like a horseshoe shape, and we seen that classical granules contain ordinary acidic phosphatase, because very active for phagocytosis. Antigen presenting cells. This is the main functions of these cells in the immune response, and monocytosis like pathological effect. Thank you very much. Our lecture is finished. I will waiting for you for our next interesting lecture about connective tissue after 2 weeks. Goodbye, everybody.